In this video, we are going to learn about secretion of insulin from secretory vesicles to the blood. I have already made a video of how insulin gets stored in the secretory vesicles. Right? So now I'm going to explain you about the secretion of insulin from secretory vesicles to the blood from this diagram, from the single diagram. This diagram is very important, right? So what here I have is, this is a beta cell of pancreas where insulin is being made. Now on the beta cell we have a transporter. This transporter is called glucose transporter, GLUT2. Now this is a 12 pass transporter. It means it passes 12 times across the membrane of the beta cell. This transporter is GLUT2, glucose transporter protein 2 transporter. Now what happened is, let's suppose you ate food just a few minutes before and the concentration of glucose is high in your blood, right? So the function of this transporter is to flip the glucose from high concentration to the low concentration. And now if the concentration of glucose is high in the blood, so glucose will bind on the extracellular area and it will be transported to the inside of the cell. It will flip inside the cell. Now glucose will convert into glucose 6-phosphate with the help of the enzyme glucokinase. Why this step is very important? This step is very important because glucose can go out if the concentration of glucose will go down in the blood, but glucose 6-phosphate cannot go out of the cell if the concentration of glucose gets low, because we have transporter for glucose but we do not have any transporter for glucose 6-phosphate. So this enzyme is very important. This is actually the sensor of beta cell. I will explain you about this later in my videos. Now let's move forward. This glucose 6-phosphate will convert into pyruvic acid and glycolysis will be performed here. We will study about glycolysis in biochemistry. Okay, this pyruvic acid will go into mitochondria and TCA cycle will be performed. TCA or Krebs cycle, we call it Krebs cycle also, and oxidative phosphorylation will also perform. By TCA cycle and oxidative phosphorylation, there will be formation of a lot of ATP and the concentration of ATP will get high in the cell and the concentration of ADP will get low because ADP is being converted into ATP. Now let's forget about this thing for a few minutes and let's move to this side. Now first of all, I want to tell you what is resting membrane potential. Resting membrane potential is the voltage difference across the membrane in the resting state. So usually for now you can just learn that resting membrane potential in usually most of the cell is minus 70 milliwatt, right? Let's suppose this is a potassium channel and this channel has a flap. Okay, this flap has two positions, where on one position where ADP can sit and second position where ATP can sit. Let's suppose first ADP was high in this cell. ADP used to sit here and keep this channel open and potassium can go out of the cell, as potassium is high in the inside the cell and less in the outside the cell. So potassium goes from high concentration to low concentration. So as soon as ATP get increased, ATP sits here and close this channel, close this flap. Now potassium is not able to go outside the cell, due to which potassium get accumulated inside the cell. There is a lot of accumulation of potassium inside the cell. Now what happen will, minus 70 milliwatt of resting membrane potential will go down to minus 60 milliwatt to minus 50 milliwatt to minus 30 milliwatt and so on. Now we have one more channel which was previously closed. This channel are calcium channel. These are voltage sensitive calcium channel. Now This calcium channel will get open as soon as the resting membrane potential will go down. This voltage sensitive calcium channel will get open and a lot of calcium will come inside. This calcium will stimulate the secretory vesicles to fuse with the membrane and release insulin, C-peptide, amylin, and proinsulin. Right? This is called exocytosis. This is how insulin gets released from the beta cell.